Hi, thanks for tuning in to the new episode of the Doctor Podcast Show, and I'm Dr. Robert Seichert, your host. Thanks for tuning in. Please click on the follow and subscribe button, and if you like this episode, and I'm sure you'll love it, please click on the like button and also the repost button, and please send us your comments. We appreciate it. Today, we're talking about a very controversial subject, something you may have not heard of before. It's called prion disease. Prion stands for a protein infection. And basically, this is a condition that you may have heard of, causing mad cow disease in England about 15 or 20 years ago. It then spread to some humans who died from it in England. And recently, we have a condition known as chronic wasting disease in the USA, which affects mostly deer. But there's some speculation recently that chronic wasting disease in deer, which is a prion disease, may possibly spread to humans and cause something called Creutzfeldt-Jakob disease, which is also caused by prions. So that's what we're going to be discussing today. I feel there's not enough attention being paid to this condition, and it could become a problem in the future because it has a very long incubation period. And so we may not be seeing the infections right now, but they may increase in the future. I'm pleased to have two of the world's leading experts on prion disease here today. We have Dr. Brian Appleby. Dr. Appleby is the director of the National Prion Disease Pathology Surveillance Center at the Case Western Reserve School of Medicine in Ohio. He's also a professor of neurology, pathology, and psychiatry at Case Western. He's also the medical director of the Creutzfeldt-Jakob Disease Foundation. He's also the chair of the professional advisory board of the Cleveland Alzheimer's Association. And we'll talk a little bit about how prion disease may possibly have some similarities or associations with Alzheimer's. And he takes care of patients with atypical or unusual dementias. We also have Dr. Krysten Schuler. She's a Ph.D. in biological sciences. She's a wildlife disease ecologist and an assistant research professor at Cornell University College of Veterinary Medicine in Ithaca, New York. She specializes in infectious and parasitic diseases of free-ranging terrestrial wildlife, such as deer, and she's an expert in chronic wasting disease surveillance and response. So let me start. Brian, tell us what a prion is and what diseases it causes and when it was discovered.
Sure. So prion starts for a proteinaceous and infectious particle. It's basically a transmissible protein. We all make the normal prion protein that has a variety of different functions in our body, but it can change its 3D conformation, and that change makes it transmissible, but also toxic to neurons. And the way it works is a process called template-directed protein misfolding, where it's kind of like having bad kids at the schoolyard that converts the good kids into the bad kids, and then they amplify and cause problems.
Great
Um,
analogy.
exactly, right? So, and in humans, it causes a variety of different prion diseases. Probably the most common is Creutzfeldt-Jakob disease, or CJD, but also it can affect a variety of different animals as well.
Right. And it was discovered in the early 1980s, the prions, right?
Yeah. I mean, really, the prion discovery started with Kuru in the 1950s. We just didn't know what caused it. It was back then. It was called, as you said earlier, a slow viral illness or transmissible spongiform encephalopathy. We knew that it was transmitted through brain tissue and caused rapidly progressive dementias, but not until the 1980s when Prusiner won a Nobel Prize for coining the term and then later isolating it. Do we truly know what was the underlying basis for these illnesses?
Right. So I want to emphasize that the prion infectious disease is different than all other infectious diseases caused by bacteria, viruses, fungus, parasites, in that all those other things contain DNA or RNA, which causes the transmission or the contagiousness. This is just a protein that's present in our body that undergoes this change, and then it changes all adjacent proteins and causes damage primarily to the brain. Is that right?
That's right. Yep. It doesn't change all proteins, just other prion proteins, but yeah, it's
Right. So we all have prion proteins in our body, but they're the normal shape and configuration. And all of a sudden, one or two of the proteins goes haywire, and then it causes that to all the other proteins, and then that causes damage to the cells in the brain. So how is this how is Creutzfeldt-Jakob disease of the brain different from other neurologic diseases that you see, for example, Alzheimer's or other similar dementias?
so all those other progressive brain disorders or neurodegenerative illnesses, they're all common in that basically they're due to misfolding of a normal protein that we create. The difference between them is what that protein is, and then what pathology causes in the brain, leading to distinct clinical symptoms. So, for example, in Creutzfeldt-Jakob disease, it's the prion protein. In Parkinson's disease, it's alpha-synuclein. In Alzheimer's, it's two proteins, A beta and tau. But within a person's brain, the same process is very similar to prion disease in that, one normal protein attaches to the abnormal protein and is converted into the abnormal form. So we've learned a lot about these other neurodegenerative illnesses from prion disease.
Right. Now, Krysten, I want to ask you about chronic wasting disease or CWD. Can you tell us the background of that and which animals it primarily affects?
Certainly. So chronic wasting disease is a similar TSE, transmissible spongiform encephalopathy, that affects certain members of the deer family. So in North America, the ones we see most commonly are white-tailed deer, mule deer, and moose. It was originally discovered in Colorado in the late 1960s, and then was identified as a TSC by Dr. Beth Williams, a pathologist at University of Wyoming, when she recognized the similarities between CWD and scrapie, which is a TSE found in sheep and goats.
Hmm. So how is CWD or chronic wasting disease transmitted? And that's the thing about these prions is you can spread the prion from one animal to another, and in the case of Creutzfeldt-Jakob disease, from one person to another. So Krysten, tell us how these deer are spreading the disease to each other.
So with chronic wasting disease, we think that most of the transmission occurs horizontally, either animal to animal through contact, such as grooming, or through a contaminated environment. And one of the challenges that we see with CWD is that the animals that are infected can live for 1 to 2 years after that infection. During that time, they look normal, but they're shedding prions in their saliva, their feces, and their urine. And those can stay in the environment where then another deer could come along and pick them up.
Right. So this stuff stays in the ground, in the soil, it's attracted to clay, and then when other deer come around and lick it or become contacted with it, they get the disease as well. And what are the symptoms that these deer and animals have? How can you tell they have CWD?
Well, it's really difficult to tell until the very end stages of the disease. And, similar to other neurodegenerative diseases, if you have holes in your brain, you're not able to function as well. So these animals typically are emaciated. They may be drooling or have drooping ears, and then eventually die. A lot of times the pathology findings are aspiration pneumonia. So basically they just can't function because is so much damage to the brain. And the brain, spinal cord, the central nervous system is where most of the prions are located. So I didn't mention in the previous answer that those carcasses that remain are a big source of prions as well.
Right. So now, Brian, going back to CJD or Creutzfeldt-Jakob disease, that can be transmitted from one person to another as well. Is that right?
But under very specific circumstances. It's actually quite difficult to transmit the disease, but basically you need infectious brain tissue, and then either it gets inoculated or put into another person's brain, like with a covering of the brain called the dura mater from a cadaver that had CJD. or it can be injected into people in large quantities. In the case when we used to get human growth hormone from cadavers, or it can be due to neurosurgical instrumentation, instruments that were used on someone with CJD that then were not appropriately sterilized for CJD precautions, and then used in another individual. But these are pretty rare instances, especially nowadays, because most of these instances have been rectified. But as again, we've said multiple times, all three of us, these long incubation periods, you still see cases, even from 45 years ago when the exposure happened. So that's the problem with these illnesses.
Yeah, there's also a case reported from a corneal transplant. I'm an ophthalmologist, and there was a case report of a patient who got Creutzfeldt-Jakob disease because a cornea was used from somebody who had that and it wasn't known. So that's a big problem. The other thing about prions is that they're very difficult to disinfect or kill, unlike bacteria, fungus, viruses, etc. With heat and certain sterilization techniques, you can get rid of them. But prions are extremely resistant to the usual sterilization techniques. Can you tell us about that, Brian?
Yeah, I mean, you need to take special precautions to completely remove infectivity, and most instances, those precautions would destroy instruments. So we usually recommend that people use disposable instruments if they're operating on a case of CJD, but you can do, special precautions otherwise, but most of the time people are just going to discard their instruments because they'll be destroyed otherwise.
Right. And what symptoms do patients have who have CJD, Creutzfeldt-Jakob disease?
Yeah, so there's a variety of different symptom presentations. It can affect any area of the brain. So any symptom arise from the brain could be could happen with CJD. The most common symptoms, though, include dementia or problems thinking. Sometimes people have an unbalanced gait, walking as if they're drunk, falling around, or not being coordinated. Sometimes people can have visual symptoms. Their eyes are fine, but the way that the brain is interpreting what the eyes see is misconstrued. And then the one that you hear a lot about in medical school is the myoclonus, which is this kind of jerky-like tremor that's very characteristic. But I've seen a variety of symptoms, psychiatric symptoms, sometimes pruritus or intractable itchiness can also be a very unusual symptom. So it's quite varied.
Quite very. And my understanding is that this has a 100% mortality rate. There is no treatment available for CJD or CWD in the ear. Once you get it, unfortunately, that's it. Is that correct?
That's correct, unfortunately.
Right. Now, tell us, I mentioned mad cow disease because that was in the news a lot years ago. Can you tell us briefly about mad cow disease and how that spread from cows to humans in England and a few other countries about 15, 20 years ago, Brian?
Well, maybe Krysten can give us a background on BSE first, and then we can talk about the human exposure.
Yeah, great idea. So, Krysten, tell us about mad cow disease and why it's called that.
I know, yeah, an unfortunate name, or bovine spongiform encephalopathy, which is a little bit larger mouthful. So there are a few hypotheses about how mad cow arose. One of them that seems the most plausible is that the disease I mentioned, scrapie in sheep, which Brian alluded to, some patients might feel itchy, and that's how that disease got its name, because sheep would scrape their wool off when they were affected. So the thought is that perhaps the cows became infected when they were fed meat and bone meal from sheep that were affected with scrapie. And this was a practice that was widely used in the UK to increase the protein content that ruminants would be fed meat and bone meal. And that was different than what we used in the US, where it was primarily from soybeans. So anyway, this TSE developed in cattle, and millions and millions of cattle were slaughtered to stop the disease. But before it was widely recognized, it did get into the human food chain, and then some younger people than what we would typically see with CJD, were diagnosed with variant CJD from eating contaminated beef.
Right. And my understanding is a few hundred people in England and other parts of the world died from that. Is that right?
Mhm.
We've
Yes.
had 233 cases to date. So,
Right. And since it has a very long incubation period, 5 to 10 years, there could still be some people who have this and don't know it, and it may show up later, right?
yeah, I mean, the mean incubation period is probably about 10 years, but as you saw with Kuru and these medically induced transmissions or iatrogenic CJD, there's a very long tail of decades, unfortunately, after that initial exposure. But the peak number is usually after about 10 years, at least in this case. We call it variant CJD in people. Um,
Right. So basically, it looks like it's under control now, but mad cow disease affecting cows clearly infected people who ate cows that were infected. That's what it appears to be. So the next thing I want to ask is, Krysten, the chronic wasting disease is spreading pretty rapidly in the USA since it was discovered. I think the last I read is about 35 states that have reported cases of it. And I think you mentioned to me that there's new cases in California, which never had the CWD. Is that correct?
Yeah, we're unfortunately California makes number 34, and that was just reported this week. So it is unfortunate. The disease started relatively slowly. You know, I mentioned it was identified in the late 70s, early 80s, in a captive facility, and then subsequently in the wild in Colorado and Wyoming. And it was really in 2002 when it was discovered in Wisconsin that people really started paying attention to it. And then subsequently, more and more states have identified the disease, which is unfortunate that it's moving that quickly. And one of the challenges that we have is that we've mentioned, you know, the carcasses can contain prions, and with hunting and people traveling to hunt or potentially moving captive cervids around, I think there's been a lot of human-assisted movement to new areas rather than just deer natural movement.
Right. And you can't tell necessarily if a deer is sick or not, because in the early phase, the deer has no symptoms, looks like a healthy deer. So if you hunt a deer and shoot it and then eat it, you may be eating infected, prion-infected deer. So my next question is there a chance that CW deer with their prions may be spreading this to humans? Because there are a lot of hunters out there who eat deer meat, which is a delicacy, venison. And are we setting ourselves up for a disastrous problem like mad cow disease, maybe even worse?
Yeah, so I guess there's a few different, you know, lines of evidence, and unfortunately, we are running a natural experiment right now because we do know of hunters that are eating known infected animals. And even though the CDC's guidance is to not, you know, if you know that your harvest is positive, to not consume it, we have the side where, you know, we just talked about with mad cow, where it did go into, you know, a smaller number of people who may be more genetically susceptible to prion misfolding. On the other side, as I've mentioned, scrapie, that disease has been known for hundreds of years, and there's been no known cases of human cases of scrapie. So we kind of have two sides of that, where we see the pathogenesis of CWD functions more similarly to scrapie than it does to BSE as far as where the prions accumulate. But it still doesn't rule out that possibility that it could occur. And that's why we urge caution and encourage hunters to get their harvests tested, especially if they know that they're hunting in an area where CWD has been detected.
Now, how do you get it tested? I understand it's a relatively long process. It's not just a simple blood test or easy test, isn't that right?
Well, because prions accumulate in certain tissues and they're in very low concentrations in the blood and the muscle. So we don't often test those tissues because you need amplification assays to find the prions there. The tissues we use most commonly are lymph nodes, so the retropharyngeal lymph nodes, kind of at the back of the throat of the deer or the elk, and then the brainstem, so the obex. And there's a limited number of approved tests for that. We're encouraging USDA to explore some of these amplification assays as approved tests so that we have faster turnaround and more sensitive testing methods.
Right. So let's say I'm a hunter, I just shot a deer. How do I get it tested to make sure to find out if I can eat it or not?
Well, most states have surveillance programs, and we work very closely with a number of states to help them design, how many animals should they test, where should they be testing. So if you're a hunter, it depends on, where you're hunting and where you live. So ideally, you will get that animal tested in the state where you shot it, and you can oftentimes the state will pay for that testing to be done, and you just have to go on their website and find out where you can submit those. If they don't have a testing program, a lot of the diagnostic labs have a fee for service where you can pay to get your own animal tested so that you are more confident in those results, in the turnaround times before you consume that animal. But we also encourage hunters to know the regulations of their home state, because we don't want them transporting some of these high-risk parts like the brain and the
Right.
spinal cord back home, where then they could introduce prions to a new area.
So what's the quickest turnaround on this test? Let's say you bring it to a center. How quick can you get a result?
Usually 5 to 10 days is
And
the,
what does the hunter do with that deer during that 5 to 10 day period? Where is it kept or stored?
well, they keep the harvest themselves. We just need the two,
So they
one of the,
refrigerate it and just kind of wait for the results?
yeah, I mean, they can process it and keep it in their freezer, and hopefully, it comes back negative, because even, there's only a few areas that have very high prevalence rates right now. So even though, 34 states have detected the disease, it's only in, small areas in each state. So
I see.
it's not necessarily everywhere. So that's why it's important to check where it has occurred.
Right. But we know that deer and prions don't look at boundaries and borders of states. They kind of go where they want. So that's problematic, and we know it's spreading. So Brian, how do you tell the difference between a deer CWD prion and a human CJD prion? Either uh, from a person who's been infected, or most cases are sporadic. Somebody just gets CJD and we don't know exactly why. Can you differentiate in your lab the difference between the CWD prion and the CJD prion?
So if we backpedal, I think we missed a very important point, and that is BSE is not naturally transmitted, right? Unlike chronic wasting disease and scrapie, where it's very easily transmitted amongst its own species, mad cow disease is not transmitted to other cattle unless you refeed it to one another, right? So basically,
Right.
you're refeeding neural tissue. So that's why they have some very specific regulations on what you can use as far as parts of the body for human consumption, but also you can't refeed animals to one another again. So
Right.
that completely eliminated BSE. The problem is with scrapie and chronic wasting disease, it's easily transmissible. In deer, it's excreted in their urine, saliva, and feces. Scrapie is a little easier because, again, these are production animals, domestic animals, but then deer are not only are they very, very infectious amongst themselves, but they're going all over the place. So
Right.
I really feel for the animal people because it's really a mess, and there's really no way to contain this like we could with other prion epidemics. So
Right.
I just want to make sure we talked about that, because I think from people like Krysten's side, this is just a nightmare. I can't imagine.
I
Um,
appreciate the sympathy.
So
But as
the
far as
question though, I wanted to ask again, can you differentiate in the lab a difference between a CWD deer prion and somebody who has CJD, let's say a sporadic case of CJD? I know you can use the RT quick test, which was worked on by Dr. Byron Cowie at the NIH. He did incredible work on that, and that's been extremely helpful in diagnosing it in people. But are there ways to tell the difference? Because if the deer prion looks like the human CJD prion, then it seems to me like it's just a matter of time before this spreads to humans and we see an epidemic in humans.
right. So those amplification assays like RT-Quick, they're very good for diagnosis, but they're not good for typing. So often in the prion field, we'll talk about things like strains or prion types. And these are more biochemical analyses like using Western blots, immunohistochemistry. So they have different biologic properties. So if I did a Western blot on a human prion, compare it to a deer CWD prion, they would look different. The problem is when you start passaging prions between species, they start adapting to other species, and then they change biochemical profiles. So theoretically, as in the case of variant CJD, which was mad cow disease transmitted to humans, those prions look different on a Western blot in a human, and they also look different underneath the microscope. So we would expect, hopefully, the same thing would happen with chronic wasting disease as well. Of course, you always want to hedge your bets and say that maybe it's not easily distinguishable. So then that's when things like epidemiology and public health becomes even more important. But theoretically, we should be able to do that. The other thing that we could do, if it's not apparent on Western blot, is we can do animal passage studies. So you can make transgenic animals that express, different species prion protein and try to amplify different prions and see what passes the most easily, determining what species the transgene is. So we would do that as well.
Right. So my understanding is that the scrapie prion that occurs in sheep, even though it doesn't seem to be contagious to humans, they have transferred that prion in the lab to non-human primates, monkeys, and also to other animals, and then it is infectious. Even though that's in vitro or in a lab, not out in nature, but it suggests that these prions can be infectious in the right environment and situation, no?
Well, I mean, I think if you make enough allowances, you can transmit, right? So you have to remember that these animal models are very specific. Most of them are overproducing the prion protein to make it much more likely to transmit. But so that's not going to happen naturally. But to your point, it does give a theoretical construct that these could transmit. And of course, with any infectious disease, you have strain variation over time and passage. So even though earlier strains may not transmit to humans or not transmit very efficiently, that could change as strains adapt either over time or through passage through other animals like cattle or, pig.
Right. Now, the prion protein is made out of amino acids. All proteins are made out of amino acids. Is the amino acid configuration in the CWD much different than in what we see in the CJD, the sporadic CJD?
Well, that's what we thought, causes the species barriers is, a difference in sequence of amino acids between the two. So.
I see. So we don't have the answer yet. Now, before this interview, I emailed both of you a couple of articles that I did in my research. As you know, there was recently a publication in the journal Neurology, which is a highly respected neurology journal. And it was based on a poster that occurred at a neurology meeting. And the authors thought that there were two cases of human CJD that occurred in Texas in two separate people who ate a lot of infected, prion-infected deer meat. They didn't have definite proof of this, but they thought that it was a definite possibility. So, Krysten, what do you think of that? Do you think that article is legit? Is there enough evidence for that? What's your opinion on it?
Well, I mean, this is sort of a correlation does not equal causation. And
Right,
so, unfortunately, this is not the first time we've seen this type of, this wasn't technically a publication because it was a poster.
right.
There was another one a few years ago out of New York with a medical student who had a poster that, sort of insinuated that Hunter ate squirrel brains and might have died of CWD when there's no link to squirrels at all. And unfortunately, it takes a lot of time for, those of us trying to explain, no, there would be a much more thorough investigation if this were, because it has huge ramifications.
Right.
So, it would have a major cascading effect on, conservation and how people feel about deer. So we need to be very thorough and very, I guess, cognizant of the, sort of the speculation that we put out there. And so the evidence, for the human transmission has been examined, as Brian mentioned, the amino acid profiles look like there's a very good species barrier. There's been epidemiological studies looking at occurrence of CJD cases in areas that have had CWD for a number of decades, like along the Front Range in Colorado and Wyoming, and they haven't seen any increase in the incidence of those diseases. So right now, we don't have any evidence of CWD going into humans, but we are constantly vigilant for that because it's an important question. Right
Right.
now, all we can say is, CWD impacts deer populations, and we need to try to minimize that as best we can. And really, we need the public to support these disease management activities to try to get those prevalences under control and not encourage further spread.
Yeah, you mentioned the squirrel cases. That was a case in Rochester, upstate New York, but there are also a number of cases of CJD, the human prion disease, in Kentucky, in people in Western Kentucky who eat squirrel brains as a delicacy. And again, that wasn't proven, but it seems very suspicious. Now, another topic, I sent you also both another article, which I found, it was published about 2 years ago, another medical journal, where there were 5 cases of CJD, the human prion disease in Michigan. That article didn't mention chronic wasting disease of deer at all, but I actually looked at a map, went on Google, found the maps, and it looks like the 5 cases of CJD in one area of Michigan overlapped very closely to the area where there was a lot of deer CWD. I tried to contact the author of that article to see if they did any studies on the people who died from that, to see if they ate deer meat as well, but I haven't been successful in contacting the author. But what do you think of that? There's an overlap of CJD cases in an area where there's a lot of CWD cases. So, Krysten, what do you think of that?
Well, the yeah, again, we don't want to jump on spurious correlations that until they're proven. With that, Michigan hasn't had known CWD for a, more than a decade. So, and Michigan is actually one of the states that has tested thousands and thousands of deer. So Kent County that you're referring to, the prevalence there is relatively low. So the timeline for somebody living in that county, which we don't know where those patients actually resided necessarily, or what their consumption habits were, it for them to get a variant CJD case from eating a deer that might have been infected at a very low prevalence, like the odds when you add those up are not great. But again, this is where we look to the CDC to try to collect that epidemiological evidence to keep working on those questions.
Yeah, I think the CDC should investigate that article some more and actually get the medical histories of those 5 patients who died from CJD and find out if they ate deer meat or not. It's pretty easy to find out. You can
So,
get their medical
if I
records
could interject here at this point.
yeah.
So those cases did come to the PRION Center, and
Oh, they did.
they go to autopsy, and we do collect information on venison consumption and hunting. Also to go back to the two hunters abstract, just to clarify a few things, the commonality between them was that they belonged to the same hunting lodge that hunted in an area that was known to have CWD. They did not have confirmed consumption of CWD infected venison. Those cases were investigated by CDC, the state public health department, and the National Prion Disease Pathology Surveillance Center. And we did not have cause of concern that they were chronic wasting disease.
Okay, so what about the 5 cases in Michigan? Do we know whether those people consume deer meat?
So I can't say anything that's not in the article, of course, because of
Right.
HIPAA, but there's nothing in those cases that make us suspicious that they're from chronic wasting disease. They reported what our final diagnoses were as far as sporadic CJD, and these are all subtypes that we expect to see in general human population affected by sporadic CJD.
Okay, so you're both experts on this. Would you eat deer meat?
Yes, I do.
You do.
Mhm.
That's
Absolutely.
interesting.
Yeah, I mean,
Where
it's I hunt in New York and get my deer tested, and my whole family consumes venison, so um, that's where we don't it it would be very problematic on a number of fronts if we stopped people from hunting, because we have no mechanism really to manage deer populations. And in many areas where we're seeing high prevalence of CWD, it's because deer densities are so high. So you would run into a host of other problems, more car accidents, other disease issues and ecological problems. So we really need to encourage that conservation message that you shouldn't be necessarily scared by this, because we don't have the evidence yet to show that it is a problem. So I worry about, like, when these media issues blow up and say that hunters died from eating zombie deer, like, when we don't have the evidence for that, because there are many other ways that people will die associated with deer problems before we actually get evidence to that. And so I'm thankful, Brian's here to talk about how they actually do the diagnosis and make sure that it is a valid result.
right. How about you, Brian? Would you eat deer meat?
I would not personally.
Okay.
Again, we have no evidence to suggest that it's transmitted
Right.
to humans, but I do think it's increasingly difficult to know whether or not your venison has been exposed. Keep in mind, a lot of these processing plants may include deer that have CWD, and it gets mixed in with others. So, again, I think that's being cautious on my part. And fortunately, I don't really like venison that much, so that makes it easy for me to say that. But yeah, but again, not because there's any link, but I think it
Right.
is becoming more and more difficult unless you hunt your own deer and get it tested, to know what you're getting.
Yeah, I think the key word there, Kristen, is you said you test it first. So should we have laws that say before you eat deer, you have to have it tested, or is that impossible to enforce?
Yeah, that would never
Won't
never
happen.
really work. And so, Brian mentioned that, some of these processing facilities, it's hard to know whether, you're getting your same animal back or if it was mixed. And so that has really been an educational campaign to try to encourage, the processors to clean their equipment between animals, to make sure that hunters, if they're traveling, that they're only bringing back deboned venison or antlers, or the cleaned out skulls. So they're leaving all the tissues that have the highest concentrations of prions in that area, so they're not moving things around. But this, like I mentioned before, even in states that have CWD, it's not like it's blanketing the whole state, except for a few unlucky ones that have had it the longest. So I think it's important to make sure that we keep messaging to hunters that this is not necessarily a scary activity, that we don't have evidence for this. And because of all the differences in the TSEs, we don't know what, whether this is actually going to jump the species barrier. So that's where I just want to urge caution that it's not overblown. And I think it's important for the
Right.
medical community to know about CWD, but not necessarily fear it as, a plague that is coming.
Right. The problem is, though, you're absolutely correct. There's no solid evidence, but because of the long incubation period, we're not going to find out until a lot of people get sick suddenly, and we're not going to have any treatments for that. On that topic, is there research being done on trying to figure out how to treat this? Do you think we're close to that, or what's the status of that, both of you, if you know?
So in
Treated.
humans, there's a phase 1, phase 2 study right now, but I mean, that's the first study we've had in human prion disease as far as a treatment trial for 17 years. So um, kind of dire.
What's the drug? Is it something that we're familiar with?
It's an antisense oligonucleotide, and the goal is to decrease the production of the normal prion protein. So essentially removing the fuel to the fire of the illness.
But the normal prion protein is necessary. It's in cell membranes and things like that. So wouldn't that be problematic?
So it's not convincing that it is necessary, and you're not completely knocking it out. You're
I
just
see.
reducing the level. It seems to be highly conserved. You can knock it out in a mouse model, meaning they don't have any prion protein, and they seem to act pretty normal. And we do know
Hmm.
that there are individuals out there that only have one copy of the prion protein, and they're fine. So presumably, it would be okay to reduce the amount of expression of total prion protein.
Right. I didn't mention earlier, but there's also a genetic form of Creutzfeldt-Jakob disease, right? It's not very common, but some people inherit this genetically where they have a defective prion protein. Isn't that right?
Yep, it's about 10% in our country.
Right. And is there a test for that? Can you test yourself to see if you have that?
Yep, you can do genetic testing.
You can. All right.
I'm
Um,
interested to hear if there's a solution to the chronic wasting disease problem from
yeah.
Krysten.
So,
Yeah.
I
Kristen,
wish I,
what's the latest research on that?
yeah, I wish I had a good answer for you. Um, Unfortunately, this is one where the pound of prevention is worth uh, more than any cure because we don't have anything that would be effective. There's been a number of vaccine attempts. They haven't really failed. I know there's a group out of Canada right now that's working on a new vaccine. But the problem that we have is, as Brian alluded to earlier, these are free-ranging animals. And
Right.
so trying to vaccinate them is, I can't even think of a way that you could do it because having seen the amount of effort and time that's put into rabies vaccinations for raccoons in different parts of the country to try to stop the spread of rabies. I mean, they drop baits out of helicopters. There's, people who spread them all over the place, and trying to do that for members of the deer family would be extraordinarily challenging and expensive. So ideally, we would like to see deer densities come down in many places and not promote congregation of animals where they would have increased possibility of transmission. So people feeding deer is a really bad idea. Feeding wildlife in general is something that we strongly discourage people from doing.
Right.
There's a lot of cultural inertia around hunting in particular areas where feeding and baiting is used quite frequently. And so trying to push back on that and explain to people why it's a bad idea to congregate these animals together is something that we try to get the messaging out about.
So you would encourage more deer hunting then to reduce the population and the population density, right?
Absolutely, yeah. It's like I mentioned, densities are very high in certain areas, and that seems to be where we have a lot of problems with CWD and no effective way to control it. But our best option is to do early management. So the first detection of the disease, if you can decrease the host density, then you stop transmission. And we've even shown mathematically that there's likely a lot of introductions of prions, and not every introduction becomes an outbreak. So having a lower density of deer is the best way to not have many get infected and then have it take off into a full-blown outbreak.
Right, but the other problem, as you mentioned earlier, is that the prions are in the ground, they're in the soil. They can stay there for months or years, I've read. So even if you eliminate some of the deer population, other deer can pick it up from the ground. So
Well, that's true. And you actually had mentioned earlier that there's certain soil types that seem to be associated with prions. The prion has an ionic charge, and it particularly likes a particle in clay called montmorillite. So with some areas that they don't have a lot of clay in the soil, the prions may get washed down below the level of the root structure where they can't be taken up into plant tissues or available to deer. And there have been instances, like New York is one of the examples, where CWD was found in two captive deer herds and also in 2 deer in the wild. And there was good interagency collaboration and immediate action on that. And there haven't been any subsequent cases of CWD detected in New York.
well, that's good. So they're figuring out how to prevent it from spreading. For people watching this, are there any websites or other online information that they can look up about chronic wasting disease and CJD? Krysten and Brian, and what do you recommend for people who want to learn more about this?
There are a lot of good resources online. The one that probably has the most comprehensive information is the CWD Alliance. So it's cwd-info.org. You can go there, and it has timelines. If you're interested, in your own particular state, it will give you information about that. If you're a hunter, it talks about what regulations are in each state. So it's a really good source. If if doctors are interested in wildlife diseases, we do have a number of fact sheets on the Cornell Wildlife Lab website.
What about CJD, Brian? Any place you recommend if people want to read about it some more?
Yeah, so clinically, I would recommend the CJD Foundation, cjdfoundation.org. If you're interested in the public health aspects or the weeds, if you will, it would be our website, cjdsurveillance.com, or CDC itself.
I see. Okay. One more question, Brian. There was a report a couple of months ago that Alzheimer's disease may possibly be contagious. They had a few case reports. Now, you mentioned earlier that Alzheimer's also has this abnormal misfolded protein or amyloid material, but up to now, we've never thought that it was contagious or transmissible or infectious. What do you think of this recent report?
Yeah, I think that study in itself is a little bit controversial, but like I said before, we definitely know that these proteins of these other illnesses propagate in someone's brain like a prion would. And there's pretty good evidence that there's a specific disease called cerebral amyloid angiopathy, which is basically when you get amyloid lining the blood vessels of the brain, that may have been transmitted through neurosurgery in some young cases. And I think most people would agree that there's pretty strong evidence for transmission between individuals for that illness. The other ones, I think, are weaker.
Yeah, so we don't have a definite answer there. I know there's been a lot of research on that over the years to try to figure it out. I think even Prusner, who coined the word prion, has done some research on that, but nobody's been able to prove it. Hopefully Alzheimer's is not infectious and contagious. Well, thank you very, very much. I certainly learned a lot today from this, and I'm feeling more confident and comfortable that two experts like you are working on this, and hopefully we'll find out more about it and prevent spreading and prevent problems in the future. I want to thank you both for taking time out of your busy days to talk about this, and it's good education for the public.
Thank you.
Thanks for having us. Appreciate it.
My pleasure.